photosynthesis. You don't have to know the minutia of, how many carbons, all that stuff, but I want you to understand, and I'm not gonna have light in there. That's a dead giveaway. Not gonna have light in there. Let me make sure I have this open. Okay, I'm not gonna have light in there. So let me go back to the review. I want you to know the equation. No, and it gets a little confusing. Don't get confused. Read it and understand it. If it has on an exam, because this is the way it is on a lot of exams, it'll call it light dependent mechanisms or light independent mechanisms. Okay? And all it's doing is talking about one phase, one type compared to the other type. It's just talking about this part, which is the light phase, and this part, which is the light independent part. And I think I might have told you in my day we call it the dark cycle, light and dark. But that's all it's talking about. The light dependent is the one that depends upon light, and the independent is the one that doesn't. Okay? So don't get confused by that terminology. One is dependent, relying upon light. The other one is not. The light is the energy. The light's the energy. Okay? The light's the energy. Okay? I want you to know what happens during both the light the light part of the reaction and the other part. So I'm going to go to this slide and talk about a lot of things, and it'll probably cover quite a bit. So here's the light portion of the reaction. Actually everything on this slide is related to the light, the light cycle, the light dependent cycle, because it depends on light. The energy from the photon particles, it's the energy from that's starting all of this actually. So the light goes in, and I'm going to talk about it sequentially, because it can happen at a lot of different places, but just for the sake of simplicity. And actually it was found that it does happen like this, but it's happening at various different steps, but it is a sequential mechanism. This is the photosystem 2. I told you the story about it being identified before photosystem 1, and then they just went with it. So anyway, light supplies the energy from that. It then is absorbed by the pigments, predominantly chlorophyll A. There's also some other light absorbing pigments. There's chlorophyll B, and that doesn't say that they don't absorb, because they do. And carotenoids, they absorb. They all absorb. But chlorophyll A is the most abundant one. So it's the principal one. So it's going to absorb that energy. It gets oxidized. That term again. It gets oxidized. So electrons get kicked off of that molecule. And in the process, electrons also get kicked off, splits water. And they literally call it splitting water. The hydrogen is splitted away from the oxygen. And the oxygen, the electrons and protons associated with the hydrogen gets, along with the other electrons that are generated from this process, get bound to a simple molecule called the primary, they just call it the primary acceptor. And this, let me back up and make one statement before I go any further. This happens in the thylakoids. This happens in the membrane of the thylakoid. Okay, in the thylakoid membrane, the thylakoids are, these sacs, and they're present in the chloroplast, and the chloroplasts are in the mesophyll cells, and the mesophyll cells are in the leaves. So photosynthesis pretty much, 99% of photosynthesis does happen in the leaves, actually, in the leaves. There's a little bit of it happening somewhere else, but I'm not gonna talk about that. Okay, so anyway, the energy from that, the initial energy all comes from sunlight. All that energy starts from sunlight, okay? And then it splits the water, it oxidizes the chlorophyll A, and then those free electrons get captured by a primary electron acceptor. And if we're going to do this sequentially, which it does tend to do, all of this happens in the photosystem number 2, which was actually the first system that was, I don't know why they call it 2. I mean, why they decided to name it 2, because it was the first one identified. So anyway, and it has an absorbance of, and it is a protein complex. It's a protein complex of a lot of different proteins, and that the absorbance for those proteins is at an absorbance of 680. And you know about a transmission absorption of proteins. Okay, so anyway, then the electrons that were captured by this process then get transported, just like with what we were talking about with our electron transport, into proteins associated with the electron transport system. So there's actually 2 electron transport systems in here. There's actually 3. There's plasmaquinine, plasmocyanine, and our good friend, the cytochromes, a whole bunch of them, just like before. So that's the commonality, one of the commonalities. And then they go through a chemoosmosis, which is just chemicals going osmosis across a membrane in the form of protons and electrons, like before, and the energy gets captured by ATP synthase to synthesize ATP. Okay, so there's also this. Now let me real quick go to this slide. And the reason I'm going to this slide is because it talks very explicitly about the electron transport, the chemoosmosis. Anytime you see the chemoosmosis, that process defines those proton-electron gradients that are generated in that membrane space, which means ATP synthase is involved. Okay, ATP synthase is involved. Okay, now let me go back to the other one, and that's the only reason I had this green one. It's from a very old text, but I like it because it explicitly says, it talks about the ATP synthase. Okay, so anyway, and then things get transferred into to the photosystem number 2, again through the same kind of thing. Ferredoxin is part of the electron transport, and for the latter part, and then you don't have to know this, but the NADP reductase will then reduce the NAD to make it NADP to make it NADPH. So here's the point I want to say about this, because I didn't mean to go through all of this. The point I want to make about this is that for this process, you have ATP and NADPH generated. That's what I want you to know. I want you to know that there is a difference between photosystem 2 and photosystem 1, but in the sequence of things, photosystem 2 is the first to absorb the light, but there's light happening, absorption at all of these photosystems, and these are in the thylakoid membranes, and all the energy that's generated in the form of adenosine triphosphate and NADPH, all of that energy gets utilized in the Calvin cycle. That's what I want you to know. All of that energy, which by definition makes this an energy consuming process. Okay, so this is why it uses energy as opposed to generates energy. It generates energy, but by the time you get to the end of photosynthesis, it's all used up. So this makes it a anabolic process, excuse me, okay? So let me go back to my, make sure I haven't missed anything. Okay, I want you to know that I've already talked about the water being split. I want you to know that the carbons that are utilized in the Calvin cycle, which is the second part of this, pretty much comes from the atmosphere. And the oxygen that's generated from the water being split, that oxygen is released into the atmosphere. And that's why people say plants make our rooms appear fresh. And that's because they do. They take something we don't need, which is the carbon dioxide from our car, from our exhaust, and they give us something we can use, which is oxygen. And this room is full of oxygen, okay? All right, I want you to know the stomata. The stomata are the pores at the bottom they're generally at the bottom of every leaf, but that's where the gas exchange happens. Now, it's also a place where water is lost, L-O-S-T. But for this portion, it's carbon dioxide going in, oxygen coming out. Carbon dioxide going in, oxygen coming out. Okay, all right. I want you to know the light absorbing pigments. We've already talked about chlorophyll being the major one. It absorbs most efficiently, chlorophyll A. But there's also a B and there's also carotenoids, okay? I want you to also understand that for our systems, and there is NAD, which gets reduced to NADH for us. For plants, it's NADP that gets reduced to NADPH. They have exactly the same function. Their function is to temporarily accept electrons and protons, you know, and they're carriers. That's why they call them carriers or acceptors, but they're temporary, and which means that they're going to give them up eventually. They're going to give up that energy, and that energy ultimately gets used by ATP synthase or other molecules in the pathway, but it gets used up. That's the point. Which by definition makes this an anabolic reaction, an energy consuming reaction. Okay. Yeah, I've talked about the light being the energy. The carbon fixation, and I'm going to go to the diagram. I'm going to go over this, but since I have it, the carbon fixation, they call it carbon fixation because carbon gets fixed. One carbon gets fixed to the next. Oh, there's also a process, I don't think I have anything on there about non-cyclic. There's also a non-cyclic portion of this reaction. This is non-cyclic. This is called linear because it goes straight from here to here. Is there anything behind there? No. Okay. So anyway, this is called linear because it goes in a straight line. But there's a cyclic one that only involves, and maybe I'll make that as an extra credit. Cyclic one that only involves photosystem number 1, where it cycles and uses ferredoxin, and it generates ATP. So it cycles and it goes like this, and it's involved in the electron transport mechanism. So ATP is generated, but NADPH is not. And that's like the lactic acid cycle. It's like a plant that will go through that if it needs extra energy for some reason or another. And the extra energy is usually some stress condition or something. Okay, let me make sure I'm going over. Okay, now there is a difference between photosystem 1 and photosystem 2. Mainly is that they're different protein complexes. You know, one, they're made up of a lot of proteins, and they're different proteins, you know, proteins that absorb at either 680 or 700. So if you look at it, photosystem 2 absorbs at 680, which means that it's a different complex, it's a different group of proteins. That's all it means. It's a different group, which means if it's a different group of proteins, it's going to function differently. They're different. I mean, it's not all that difficult to grasp. If they're different group of proteins, and I don't know which proteins are in common or not between them, but because they have different complexes of protein, they absorb at different wavelengths. That's all that is. Okay, all right, we're almost done. We're on the downside of this, so just bear with me, probably another 10, 15 minutes. Oh, or bye. Okay. Oh, no, the end product of the Calvin cycle. Oh, actually, we only have two more things. The end product of the Calvin cycle is G3P, the ceraldehyde 3-phosphate. It is not glucose. Glucose is made later. At the Calvin cycle, at the end of photosynthesis, it's ceraldehyde 3-phosphate. And those get together, two of those get together to make a 6-carbon glucose or whatever, however large and all that stuff. Are y'all with me? Okay, good. Because even though Mary's leaving, we're about to be right behind her. Because we're nearly done as well.
Wait, how long?
Probably about another.
I can say another 10 minutes.
Actually, I think it's done. I think we're done. No, this is the end. This is the end. This is the end product of the Calvin cycle. Now, what do y'all need as clarity? Because otherwise, I'm gonna close this room up and I'm gonna run up to the lab. How y'all doing? Valerie, how's it going? Absolutely. The cytochromes. What else?
What?
Let me do Valerie right quick. Okay. Now here's what the cytochromes are proteins, and they're involved anywhere where you see CYT. Those are cytochrome proteins, and they're part of this electron transport right here. See this? So they're accepting electrons from NADH or NADPH for plants, and FADH for us, okay, or FADPH for them. Are you with me? So they're just proteins, and I should have made sure that I set that in class, because all I just pointed cytochrome, that's it. But these are the cytochromes, these are proteins. The CYT, that's abbreviation for cytochrome. Now they're part of their temporary carriers, just like these other molecules. So they're temporarily carrying the energy around, okay? Now, there are also cytochromes associated with plants. It's probably, I don't know about every time I get to this point, I say, I'm gonna compare the sequence for these. It's very simple to do. Or I might have it as an extra credit next year. Well, actually, I'm no longer teaching freshmen. But do you see the point? The same cytochromes associated with proteins, with plants as well. Are we clear? Now, should I go to the other slide and show you the cytochromes on the protein, on the plants? Remember, if I go to, let me hold on one second. Hold your horse, Mary. You're just chomping at the bit. Oh, here, right here. Here it is, right here. It's right here. See the cytochromes right here? Cytochrome complex. But I know this is a lot of stuff to remember, especially if you've never seen it before. Are we good? So it's cytochrome.
So it goes from cytosystem number 2 to cytosystem.
I don't know why they did that.
Can you just repeat the end product of the photosynthesis?
The end product, the last molecule that is made in photosynthesis is G3P.
G3.
Let me show you. It's on your slides. And it is and I'm glad they just, because usually I have to. What's not on here?
G3P.
Where?
Right there on the
It's on the far upper right, yeah.
Oh, yeah. But I thought I thought it was designated somewhere else. Okay, so anyway, G3P. At the end of photosynthesis, this is the end right here. This is the end, this is the end. And then, so you have the G3P, which is the glyceraldehyde 3-phosphate. And then that's glucose and other molecules are synthesized later. But officially it is a sugar molecule. What's your question? You look like you had a question. You sure? Okay.
Just carbohydrate.
Thank you.
Which one?
It said, how to cycle the product is, 3-phosphate.
Oh, glyceraldehyde 3-phosphate. That's G3P. Now, when on the exam, when I have something like an abbreviation, I'm gonna give the abbreviation and the name. Oh, I'm glad you mentioned that. I'm looking at your name, but I'm not pronouncing it, so I'm not gonna say it. I'm looking at it, but I'm not gonna say it, because it's gonna make you mad. Okay, so what goes in, there's individual carbons bind to RUBP, ribulose bisphosphate. So going into the Calvin cycle, so remember I said I want you to know what goes in and what comes out. What going in is you have a 1 carbon, there's 6 of them that get together, but individually is 1 carbon binding to a 5 carbon RUBP. Okay? And when I give it to you on the exam, I'm gonna have RUBP/ribulose bisphosphate. Okay? So, and that's what this is. And this it's 1, 2, 3, 4, 5 carbons bonding to that 1 carbon. So, and it's 6 of those. 6 of those bonding to 6 of those. But what's important for you to know is that you have, and they call that carbon fixation. That's what they call carbon fixation, because that's fixed. 1 carbon bonding to another carbon, bonding to another carbon, bonding to another carbon. So this process is called carbon fixation. It's called carbon fixation process. Okay? So it's fixed, and it's actually binding to it. It's a covalent bond generated. So it's a carbon fixation process, but it's RUBP binding to an individual carbon, and at the end, G3P. And on the exam, I will tell you ribulose bisphosphate, parentheses RUBP, glyceraldehyde 3-phosphate, parentheses G3P. So you don't have to memorize all those acronyms. Okay?
While the photosystem number 2 is the first to absorb the light and stuff.
Yeah.
What is the photosystem?
Same thing.
Same thing.
Oh, yeah. Now, here's the point. I'm glad I want y'all to get all these things. Now, it's going to, now let me, hold on 1 second. I'm going to go to this diagram. So think about this as being what they call a linear procedure. Linear just means in a straight line. So in the sequence, it doesn't mean that it's not happening, light's not absorbed all it, but if we're looking at it in a sequential way, which it does happen, you're gonna have photosystem number 2, you're gonna have the light coming in. Let's go through the process. You're gonna have light coming in, and it's gonna be, it's the sole source initially of the energy. It's gonna be the only source of the energy that's coming in is coming from the light photon particles. So that's the energy. That's why it's in the equation. So it's going to oxidize the chlorophyll A, kick off electrons from the chlorophyll A. And then that same energy is going to be used to split water in two. To split water into hydrogen and split water into oxygen. Are you with me? And then that same energy is going to be absorbed by another molecule called another acceptor. But and then that energy is going to be transferred as hydrogens to all these molecules. So it's going to become cytochrome with an H, plasmocrinone with an H. Do you see the point? Are you with me in that? Yes or no? Okay. And then that all that hydrogen, those hydrogen protons and electrons are going to be used by AT synthase to make ATP. Are you with me? And then it's going to transfer to photosystem number 2, and then there's going to be another electron transport, and NADPH is made. So it does happen sequentially. It does happen sequentially, but at the same time, I don't want you to lose sight with when the light comes in. It's not just going to hit this photosystem right here. If it hits this photosystem right here, I'm not sure what will happen if it just hits this and not here. Do you see the point? So the photosystem number 2 is part of this sequential transfer of energy. Think about it that way, because it is a sequential transfer of energy, transfer of energy, and the end of and actually photosystem 1 is very important for another reason as well, because photosystem 1 there's an electron transport mechanism that specifically generates NADPH.
So the second system breaks it down to create ATP, and then it goes to the first one to make
Yes. Now they are different. They're different systems. You know, and somebody genius named. I went back because I was interested when I first started doing this. So why would they name this? Why would they named photosystem 2 before the first one they first identified it, they just named it 2, photosystem number 2. I don't know why. That just doesn't make sense to me. And then later on, they make the discovery, oh, there's another photosystem. They said, well, backtrack, they just named that photosystem 1. So, I don't know.
Why did they just switch it?
Mainly because they probably had it published like that.
So everybody just already has it stuck in their head that it's
You're
photosynthesis
stuck.
too.
Actually, you get stuck with things like that.
Wow.
You get stuck. And it's not catastrophic. It's just interesting that that's how it happened. But I want you to know about the water being split, which is very important. I want you to not so much this whole sequence, but I want you to visualize it and understand it. But water gets split. This is the light part of the reaction. Water getting split is a key component, a key character, a key responsibility of this whole process. Water gets split, you know. And then the other part is that energy is generated, adenosine triphosphate and NADPH. Those are 2 critical processes related to this. You know, the ATP, the water split, the ATP and the NADPH, and all of that energy, all of the energy that's generated, because overall this is an energy requiring mechanism. It requires energy. So if we were just to stop at this point, we'd say, okay, it's an exergonic reaction, but it's not, because the energy, all of that energy gets utilized. All of that energy gets used in the Calvin cycle to put those carbons together. It takes a lot of energy, and they call it carbon fixation, but you're actually bonding individual carbons together. Individual carbons, you're bonding them together. And it takes energy to make a bond. You get energy when you break a bond. Okay? It takes energy to bond things together, to generate covalent bonding. It takes energy, and when you break bonds, you release the electrons in the outermost shell, the things we talked about in chapter 2. Electrons in the outermost shell have the most energy, and when you break bonds, you release that energy. You release the energy that is the most carried. According to the physicists, they've determined, they've found that the electrons in the outermost shell, which get broken because you have one orbital and another orbital, and all it's seeing is the electrons on the outermost shell. So when you break those bonds, you release all that energy. You release all that energy. So that's what happens when you eat. When you eat, you're breaking bonds. That's what digestion is. Digestion is breaking bonds. Because you're breaking up all that food. Okay. How are we? Are we good? Okay, then let's go.